president here at Wake Audubon. Thank you all for joining us. We're going to try and run a timely meeting tonight. So if you're interested in the debate, you might miss the beginning of it, but at least you'll get to see most of it. I have a few announcements before we get started with our guest speaker. So as probably many of you know, we have bird walks most weekends in the area. So this is like maybe a little too much. I'm gonna turn this off. Can you guys still hear? No?
I can hear.
I think this is interfering with this, John.
No, you're doing fine.
So you can check out our calendar on our website to see when our different bird walks are. And the best way to find out is to look at our meetup page and join our meetup group. Most of our bird walks you can RSVP through the. I don't know where the interference is coming from.
I can turn this off and then it won't be. Oh, is it? It might be coming through two different.
I'm not on Zoom on here.
Okay, it might just be coming through now.
All right, I'm going to speak up. So you can RSVP through our meetup page. So just as an example, we have walks coming up at the Wendell Community Park, Durant Nature Preserve, Bass Lake, Hilltop, Needmore. We have two chimney swift viewing events coming up. So the first is on September 24th. It's going to be at the Willard Hotel, which is in the Glenwood South area. And John, an ornithologist here at the museum and also on our board, is going to give us a talk from 6 to 6:45 at the hotel. And then we'll go out to their rooftop patio and wait for the swifts to show up for the chimney that's next door. They normally come in just right after it gets dark, and there's drinks and food on the patio. So, and then we have another one a few days later on Sunday, September 29th. It's at Broughton High School, and you can, again, 6 to 7:30, and you can show up there. If you don't mind, RSVP to John Connors, and we'll drop his name in the chat. And you can also find the information to contact John on our calendar or meetup page. We have some volunteer opportunities coming up as always. So we have a work day coming up at the Prairie Ridge. It's the Prairie Ridge Eco Station. This is something we do on a semi-regular basis to help maintain the area. So we'll be like weeding beds, picking up and removing debris, pruning trees and shrubs. If you're, I don't think Robin's here tonight, but if you're interested, again, you can RSVP on our meetup page, or you can contact Robin and she can get you set up. We also have lots of like leadership volunteer positions that we're always trying to fill. So we need like a volunteer coordinator. We need somebody to be our conservation committee chair. We need someone to help us with like tabling materials. If you are interested in that or other volunteer opportunities, come find me and we can chat. Or you can email me in the address that will drop in the chat. And then tonight is the start of our lights out season. So as many of you all know, keeping lights on can confuse the birds, and there's an increase in bird collisions during this time of year, during the migration. And so we ask people to turn their lights off from 11 p.m. to 6 a.m. between now and November 30th every night. And we have partnerships with the city and others to like help the city's parks department to help promote this. We also have a pledge online that you can sign to show that you're going to commit to do these actions and support us. And if you follow us on Instagram, we'll be having posts throughout the next like month or so all about lights out, and you can learn more. And then the last thing is, if it is your first time joining us here at Wake Audubon, we will have a special deal that we always offer new folks. We will pay for your membership in Wake Audubon for the first year. This includes membership in the National Audubon Society. So if you're interested in that, come find me after the meeting and I'll get your information and we'll get you signed up. And with that, I'm going to turn it over to John to introduce our speaker for tonight.
Well, it's nice to be able to introduce our speaker because we first met back around 2018 and then kind of lost touch. So when Robert invited me to East Carolina, where he was a graduate student, where I gave a talk, and later we got Brian to go down and give a talk. But now it's nice that we're inviting him back to give a talk here in Raleigh, Wake Audubon. So I'll give you a few bits of information. You got his biological science. Oh, right, we're online. I forgot. And all right, start over. Robert got his 4 years BS degree at Muhlenberg College back in 2009 and 2017. He went on to get his master's in biology at Villanova. Then in 2022, he had another degree, his PhD at East Carolina University. He is now an NSF postdoctoral fellow, research fellow in the Department of Molecular Genetics and Microbiology at Duke University. He is now in the lab studying bird olfactory receptors, bird sense of smell. I'm sure you're thinking, wait a minute, birds don't smell. Or most birds don't smell. But we'll learn about things that do and don't. Now, the trick question. So only recently has he gotten into smell, but for years he's had a favorite bird. What do you think that might be?
Vultures.
Vultures. How about that? It's like he's come full circle in his life where he's now studying a bird that has great olfactory. All right, well, please welcome Robert Driver. We have to share the screen. We'll get that going first. All the little tech stuff. Yeah.
How's it look on the Zoom?
How's it look out there? Oh, there they are. Do you want to use this or the other?
I think we're
I'm
seeing
sure.
presenter mode.
They're seeing presenter mode. So you guys are.
Switch the screen.
Presenter. We can get out of presenter if you.
Yeah, I don't need the present. Still a problem, right? Oh, we should have tested this before.
There's some little way to get out of it. There's a couple of other dots you can end presenter. And whoa. Use the slideshow. It said use slideshow.
How about now?
There you go.
Yeah,
Yeah, now it's good. All right.
Try
Thanks,
that.
John.
Try that mic if it doesn't work.
Okay. I'll try this mic. I'll try to stay close to it. All right, everybody. So, okay. Let's turn the lights. Oh, yeah, this is very relaxed setting here. Yeah, thanks everybody for coming to my talk on bird smell and bird olfactory receptors. So I don't always talk to a bird audience, so I just wanted to just open it up. Oh wait, can I get rid of this actually? Yeah, I just wanted to ask, does anybody in the audience or at all have any thoughts about what they've heard in the past? Can birds smell? What have you heard about it? What do you know about the sense of smell, or do you have any observations? Anybody heard things? Yeah. Yeah.
Yeah, they smell if you don't change their flavor.
Yeah, that's correct. Yeah. So just a clarification about this slide, referring to the sense of smell. as opposed to the, yeah. But anybody? Yeah.
Some vultures.
Yes, yes. So some vultures have a heightened or have a specialized sense of smell for sure in their foraging.
And that particularly turkey vultures?
Particularly turkey vultures, yes. And turkey vultures in the vultures in the genus Cathartes, yeah, the yellow-headed vultures. So if no one has additional thoughts, that's kind of consistent with what people have thought about bird smell in the past. And trying to advance here. And so one thing that we all know about birds is that they're very brightly colored. Some species are very brightly colored, many different colors in the rainbow. They have elaborate ornaments coming off their tails. They have these sexual displays that they perform, and many of them sing beautiful songs. And so it's obvious, pretty obvious that vision and hearing are important to birds. And this has led to prevailing thought in ornithology. The birds are mostly diurnal, they're often colorful, they sing these songs, and so therefore they must rely on vision and hearing at the expense of olfaction. And this idea was really synthesized in this 1905 Nature paper from Alex Hill, where he looked at morphological and behavioral evidence in the turkey, and he concluded that olfaction was not very relevant to bird behavior and ecology in his paper. And it was supported by earlier experiments by some most famous biologists ever. John James Audubon, he took a deer carcass and he buried it under the ground. And then he found that the turkey vultures couldn't navigate to it in this particular experiment that he performed in 1826. And if you read Voyage of the Beagle, there's a part where Charles Darwin is lined up in front of this row of Andean condors, you know, this huge, largest flying bird, and they're chained up, and he's got a piece of meat wrapped up in paper in his hand, and he's showing them all the meat as he's walking in front of, or showing them paper as he's walking in front of them, and they're all just kind of standing there, just standing there. And then he rips open the paper and he shows them the actual meat, and they all start fluttering like crazy on their chains trying to get at it. And he concludes, I couldn't have fooled a dog this way. He concluded that they were using vision, not smell, in their foraging based on that little experiment back in 1800s. So this led to a real delay and lack of attention to smell in birds for a long time. And it wasn't until the latter half of the 20th century that we had the first experiment that showed that birds could smell. So in this 1966 science paper, they trained pigeons to peck a keyhole here. And when they peck the keyhole, they receive a seed. So they trained the pigeon to know to peck this keyhole and receive a seed. And then they started adding amyl acetate to the air behind the keyhole. And anytime the pigeon entered the keyhole with its beak while the amyl acetate was flowing, it received a shock. And so it learned that the amyl acetate it shouldn't go into the keyhole when the amyl acetate is flowing. And so they adapted to this behavior. They stopped trying to access the seed when the amyl acetate was activated. And this was the first controlled experiment showing bird smell in the pigeon, it was. And then since then, in the past maybe 20 years, there's been really an explosion of studies showing behavioral evidence for bird smell. And I'm just going to highlight maybe one too many here. I have a few slides on this, but some of my favorite ones. So speaking about how birds smell, like the paper on your parakeet or whatever species it is that you might have. If you pick up a crested auklet, I don't know if anybody's really done that, but and you sniff it, it smells like citrusy, like an orange. Actually, I'm talking to bird people, so maybe someone has done that here. So, and they had this very citrusy like scent. And so what Julie Hagelin in this 2007 paper did is she set up tea mazes where the crested auklets had a choice to go to one side or another. And one side, for example, had fresh crested auklet feathers in it. while the other was just a control with nothing in it. And you can see on the black bar has the feathers. The majority of the time, the crested auklets went to the side with the feather smell as opposed to just a control. They also went to the side with a synthetic mixture that contained certain compounds within the crested auklet feathers and a control of banana odor. They didn't have a preference, and they actually avoided a predator odor a little bit, showing a species recognition component to smell in birds. And then for individual recognition, this was a cool paper that came out where they took zebrafinch eggs from the nest of the parents and zebrafinch eggs that were not from the nest of the parents and put them behind these little fans and had the fans blow into two sides of a cage that had individual chambers in it. And they found that although there was no difference after day 3 after laying an egg, after day 10 after laying the egg, if you put the same finch's egg on one side and a different finch's egg on the other side, the finches prefer being in the chamber that has the smell of their own egg. And so this was an example of individual recognition for smell in birds. And just a few more, and then I'll stop going over these. But we could do it all night, and then you guys would may miss the debate. But the starling here, they put out unscented nest odors and also nest odors with vanilla or this milfoil seed-like odor. That was dipped in the nest materials, and they found that the starlings preferred to select the vanilla or milfoil smelling nesting material over material that had no odor. So this was a nest material selection example for olfaction. And I think just two more here. But one of my probably my favorite example of olfaction in birds is for foraging. And so one of the well-known groups of birds for smell are the Procellariforms, the pelagic species. And so they're navigating over this open ocean with no landmarks, no real visual cues for distinguishing where they are. And so Gabby Nevitt at UC Davis has done a ton of work on Procellariform smell. And in this PNAS paper, in the top left, they laid down some oil slicks for the wandering albatross to navigate to. And the bottom right is where the albatrosses started, many kilometers away from where they deposited the oil. And the wind is going this way, down. So the albatrosses are downwind, and they're able to fly and navigate to the oil slicks. And it was concluded that this flight pattern was indicative of using olfaction as a guide. So certainly Procellariforms using olfaction. And then this paper, which I really like, that came out just a few years ago, started from an observation that in Europe, when they mow the lawns, there's the storks that just appear near the mowers, like in that picture there. And so they were curious, do the storks smell the freshly mowed grass? And so here's an example of this one was mowed, and the wind is going this direction, and the stork is able to come upwind as it smells the mowed grass moving downwind. And so they showed this in a semi-controlled experiment, I guess, where the blue dots are just water that they sprayed on like a negative control plot. And the green, they sprayed the leaf alcohol, which is supposed to simulate the smell of freshly cut grass. And the wind direction is the blue arrow going down. And the dashed white lines are storks that didn't move, just showing the stork's position relative to the different plots. But the yellow arrows are storks that did move towards the plot. And the only direction that they will move, the only time that they will move towards the plot is when they start downwind. And so that was the only direction that they navigated towards the plot. And the authors concluded that the storks were using smell to find these freshly cut lawns. So that was all really cool. And of course, I want to mention the three other groups of birds that have been shown to have highly specialized foraging. So kiwis, I'll mention a little bit later. Of course, they're almost blind. They have their nares for breathing at the end of their beak, unlike every other bird, which has it at the closer to the head. And so they probe the ground and look for worms and invertebrates that they're foraging for. And then honey guides in Africa. There's some really cool old studies showing that they're attracted to lit candles. And so they are attracted to wax, and it's thought that they use olfaction to find the beehives that they need to navigate to. And then Cathartes vultures, as you guys have already mentioned, actually have an excellent sense of smell. You can see their perforated nostril there, which is really adept for finding carrion under the forest canopy. And it was thought that John James Audubon was using carcasses that were too rotten for the turkey vulture in his 1800s experiment. So that's some great information about bird smell behavior, but what's really going on in the sense of smell? So just to so everybody understands this, the sense of smell. So you will breathe in air, which will have odor molecules in it, and then they'll come up through your nose. And in humans, on the roof of your nasal cavity, there's this little bit of tissue called the olfactory epithelium. And there's neurons here, which will take up which will convert the odorant molecules to a signal for your brain to perceive. But at the interface between the odorant and the olfactory epithelium are these cells right at the tip where the air flows across, called the olfactory sensory neurons. And on the cilia, at the tip of the olfactory sensory neurons, are the olfactory receptors. And these are the receptors in all vertebrates, you, me, birds, lizards, whatever, that will actually bind to the odors. And it's the interaction with the environment in the sense of smell. And so this is an olfactory receptor here. There are these proteins which are found on the membrane of the cell. And so the odor comes in, so the olfactory receptor is just the pink part, and the odorant, in this case propionate in this cartoon, comes in, binds to the olfactory receptor changes shape, it sends a signal in the cell that eventually goes to the brain and allows you to perceive and then eventually respond to that odor. And so there are many, many different odors out there, so many different kinds, all different shapes. They can have be polar, non-polar, have a sulfur, have a ring, be large, be small, be charged, not charged. So it's in this paper, I'm not really sure exactly how accurate this is, but in science, they claim that humans can perceive over 1 trillion different odors. So huge number of odors to perceive. And in order to accommodate this, olfactory receptors work in a combinatorial fashion where any given odor can activate one or more than one receptor. So in this cartoon example, this is a 5x5 grid where each square is an olfactory receptor. Let's say there's 25 olfactory receptors in this grid. Here, this odor activates one olfactory receptor as shown in red, but a different odor can activate 2 olfactory receptors. And this olfactory receptor, one down on the second row all the way to the right, is activated by multiple odors. So an odor can activate multiple olfactory receptors. An olfactory receptor can be activated by multiple odors. Some odors can activate many olfactory receptors. And some odors in the blue here in the bottom right can activate some olfactory receptors and deactivate other olfactory receptors. So this is how you can get to like 1 trillion different odors combination, because there's just so many things going on with all the olfactory receptors. And so to accommodate all these odors, olfactory receptors are the largest gene family in all of vertebrates. And so some species have over 2,000 olfactory receptors, and these counts are a little bit outdated, but in some recent papers, they showed that elephant had, both Asian and African elephant have over 2,000 olfactory receptors in their genome. Dog there has 800, mouse with a little over 1,100, and us humans, we have about 400 olfactory receptors. And smell is, of course, important to all these mammalian species. But what about in birds? Is olfaction important to birds? So we saw the behavioral examples that it's important, but can we get some molecular evidence from the olfactory receptors to support this? So we saw that mammals have a lot of olfactory receptors, but do birds have a lot of olfactory receptors? So to do this, we need to go into the bird genomes. All the birds the complete bird DNA. And the way we do this is it's basically like almost like using Google. We have a set of known olfactory receptors, a file of known olfactory receptors that are already published from mammals and reptiles. And we use that as our search query. And we search the bird genome, in this case, the chicken, with that search query, and we recover the hits, and then we filter them, and then we align all the sequences, and then we build a tree, which is this. And this tree, each tip in this tree is a single olfactory receptor showing how closely related they are to each other. And every time we do this, the tree always forms this shape, this stereotypical shape, almost always. And the shape is created by the three groups of olfactory receptors in birds, which are the alpha, down here in green, the gamma, here, this is alpha is for water, detecting waterborne odors, the gamma here in yellow for airborne odors, and then the gamma C in blue there. And the gamma C are very special. They're the vast majority of bird receptors are gamma C. They have these short branch lengths, so very high sequence similarity, and they're only found in birds. They're not found in any other vertebrate. They're not even found in crocodiles or the most closely extant relatives of birds. So a group of olfactory receptors only in birds. And so we found when we scanned the chicken genome here, we found that the chicken genome had 473 olfactory receptors. So not only do birds have olfactory receptors, the chicken has even more than us humans. So now we can do this across the whole bird phylogeny. So we looked at, as of right now, about 177 different species of birds. We found over 44,000 olfactory receptors total. The average is about 253 olfactory receptors per species, but the repertoires vary very widely from 7 to, we'll get into the high examples. And you can see 80% of those are the gamma C receptors. So we'll talk about the highest ones here that we found. So not only can mammals have over 1,000 receptors, but we find over 1,000 receptors in different pigeon species. So the Nicobar pigeon and the band-tailed pigeon there both have over 1,000 receptors. The ducks consistently have very high receptor counts, with mallard at almost 1,200 there. And then we have 3 pochard species, which are all around 1,000. Showing that ducks just have a high count of receptors. But then the kiwi, okay? 3,750 olfactory receptors. When we first started looking at olfactory receptors in birds, I thought maybe we'll find some, maybe we'll show that they were equivalent to mammals. I didn't think we were going to find the animal with the most olfactory receptors of any vertebrate, of any animal. And that's the kiwi, which just blows the elephant out of the water. And so, which is hard to do, an elephant's so heavy. And so if we could just appreciate it for a moment, this is what the kiwi genomic olfactory receptor repertoire looks like on a phylogeny, where just the gamma C alone are greater than the total number in the elephant. And there's all these short branch gamma expansions. It's just a fascinating repertoire that the kiwi has, and it's consistent with its morphology and behavior that we know. And then on the low end, there's actually some species. If I could move this, maybe I can move this thing. Maybe I can't move it. There we go. Maybe it's better over here. Anyway. So the lowest counts were largely in songbirds, although there's a woodpecker there. And the New Zealand wren had very low counts, but consistently we have 4 crows in the data set. Crows are the lowest of all birds that we found so far, and that's pretty consistent. And we didn't expect this result. We sort of thought maybe crow is a generalist forager, but we cannot find more than a dozen or so receptors in the crow at the other end of the spectrum. And these are some of the lowest counts of all vertebrates, similar to toothed whales, which have lost the olfactory organs altogether. So we have low counts and high counts when we look across the bird phylogeny. So do birds have a lot of olfactory receptors? Yes. So there's often hundreds of olfactory receptors, but it's dynamic across different bird species and is changing. But we want to know, are the bird olfactory receptors expressed in the olfactory epithelium? So we got the genomic counts. We got the counts of the olfactory receptors from the genome, but every cell in your body has the same DNA content. So by getting the genomic counts, we don't know where those olfactory receptors are actually functioning or playing a role. So if you look here this is a study in humans, each row here is a different olfactory receptor, and each column is a different tissue. And you can see the different tissues, I mean, whatever you want to name, heart, kidney, liver, lung, they all have olfactory receptor expression in them, illustrated in blue. So the darker blue, the higher the olfactory receptor expression for that tissue. So birds have olfactory receptors, but olfactory receptors can be used for different things in the body, and we want to know, are they being involved in smell? And so to do that, we have to get some olfactory epithelium from the bird. And this is my little shout out here. We didn't know how to get the olfactory epithelium in the bird. So we had to perform this unique dissection where we open up the maxilla, the upper mandible in the bird, and go fishing around for the olfactory epithelium, which we did find here. You can see there's these like spiral-like structures, which are the turbinates of the olfactory epithelium used for temperature modulation of the air that's being inhaled. And so Brian was really nice to let me into the museum a few years ago and practice these dissections on a bunch of different birds. Here I'm doing it on a killdeer and a green heron and then a scoter, and really helped me learn about where the olfactory epithelium is in the structure inside the maxilla. So after tearing through these birds a little bit here at the museum, we then obtained olfactory epithelium and pectoralis muscle as a control from 4 different species. So we got a chicken from NC State right here. It has the Department of Poultry Sciences. I don't know if any of you have been to this chicken farm that they have there. But then zebra finches from Duke, and cowbirds from UPenn, and then Anna's hummingbird there from UC Riverside. We obtained the tissue and measured the expression of all the olfactory receptors. And when we did that, we found that not only were some olfactory receptors present in the olfactory epithelium, but basically the entire genomic repertoire was there. So again, here is the genomic repertoire where each tip is an individual olfactory receptor. And then here we have the expression of the olfactory receptor in the olfactory epithelium for this column and the pectoralis control muscle for this column. And yellow is low expression, purple is high expression, and black means it was absent. And so the olfactory receptors, the majority in the zebrafinch, every single one of them was, but the majority of olfactory receptors are expressed in the olfactory epithelium, and there are more expressed in the olfactory epithelium compared to the pectoralis control muscle, and they are often more highly expressed in the olfactory epithelium than the control pectoralis muscle for the ones that are expressed in both. And when we looked at this across the phylogeny of the 4 different species that we have samples for, we saw that this pattern was consistent, showing that the olfactory receptors are expressed in the correct tissue. So this was a good sign, but the olfactory epithelium has lots of different types of cells in it. So there are, this is a zoom in of olfactory epithelium from a song sparrow, from this paper in the Auk a few years ago. I really like these images, and as well as a picture of the olfactory epithelium from Leach's storm petrel. And there's these curled turbinate structures that I was talking about before. And in here, there's many different cell types. There's basal cells. There's these type of cells called Bowman cells. There's progenitor cells. But the cell type that we're really interested in is the olfactory sensory neurons, which I mentioned at the beginning, are the cells that have the olfactory receptors at the surface of the olfactory epithelium. And so what this looks like in the mouse, this is a mouse olfactory epithelium in the top left. And this band here, sorry, the people online can't see me pointing, but this band here is the olfactory sensory neurons in a mouse, which are located on the perimeter of the curling turbinate to interact with the air coming in from the environment. So we were looking for a similar pattern in the chicken. And so we had to find the olfactory sensory neurons because we didn't know where they were. And then we want to ask, are the olfactory receptors present in the same location as the olfactory sensory neurons? So the first thing we did was sample from the front of this is the song sparrow, but I'm talking about the chicken. We sampled from the front of the chicken's beak and we couldn't find anything. Then we sample from the middle concha of the chicken's beak and we still couldn't find them. But then we finally sample from the posterior concha of the chicken's beak, and I'll show you what we got. So this is the posterior concha of the chicken, and that's the curl of the turbinate there. And this is a nuclear stain where each dot here is a cell's nucleus in the chicken posterior turbinate. And our first probe that we're using is this thing called CNGA2, but you don't really need to know about it. It's just a positive marker that indicates the presence of olfactory sensory neurons. This is a protein that is only present in the mature olfactory sensory neurons, and so it can tell us where the olfactory sensory neurons are. And when we use this probe, we see that the distribution is just as we would expect along the perimeter there of the posterior turbinate, just inside from the outside, like we saw in the mouse. And then when we do the same nuclear stain, but this time we probe for the actual olfactory receptor, in this case gamma C, we see the same distribution of the gamma C olfactory receptor being present right along the perimeter in the same distribution as the mature olfactory sensory neurons, showing that not only are the olfactory receptors present in the correct olfactory epithelium tissue, they are present in the correct olfactory sensory neuron cell type in the birds. So this is all pointing in a very good direction for bird smell here. So are the birds ORs expressed in the olfactory sensory neuron? Yes, they're expressed in the olfactory epithelium. Yeah, sorry. When? Like what date? This year. Yeah. This was in April. Nobody had looked at it, no. Although it took 8 months to get that. So it was also a little bit of last year too. So yeah, no problem. Yeah, sorry if we enter, you can ask a question anytime. Sorry. I just get so into it. You might have to raise your hand and shake it a little bit. Yeah.
So the very first, I get the anatomy pieces, but when you were saying that it was the genes you were expecting to see were expressed in the tissue, how do you know those genes are expressed?
Oh, yeah. So okay, so right, so for this one or for this one?
Yeah.
Oh yeah, sure.
So how do you know like what is on the right is matching like what's on the left, essentially?
Yeah, so these are mRNA. So the question is, how do you know what's expressed? Is that the question? It's like yeah, like how do I take a transcript and assign it to one of these tips? Like okay, so we get the mRNA sequences and then there's a software which will determine can the sequence be accurately mapped to an individual gene or an individual tip. Some of the sequences are a little ambiguous and it can't be accurately mapped to a single tip, and so they get thrown out. But any ones that can be accurately assigned to an individual a specific olfactory receptor, then contribute to the count that's associated with that tip.
And that essentially means that the gene in that genome is like turned on.
Yes, so these are sorry, sorry, my fault, guys. Yeah, so these are mRNA measurements, not the DNA. So anytime you get an mRNA transcript, it means that the cell had to transcribe that from the DNA, and the assumption is that that's going to be translated and then expressed. So yeah. Yeah, it turns on the gene. Yeah. So, okay, cool. Right, and this is also using mRNA probes. So this is also measuring the mRNA present, not the genomic DNA. So this is like the gene is turned on. I'll remember that for next time. No, sorry, guys. I did do the thing in my mind, like your every cell has the same genetic content. I prepared that at least. Okay, so This is great that they're in the correct place, but what we really want to know is, do the bird olfactory receptors respond to odors? And we can ask this in two ways, one for the gamma C and one for the alpha receptors. So to do this, we have to perform the first functional study ever in bird olfactory receptors. And the way we do this is, there's too many olfactory receptors. We want to match the olfactory receptor to an odor. And there's too many olfactory receptors to test because there's hundreds of them. So the way we do this is we create something called a consensus olfactory receptor. This is where we take a group of olfactory receptors, in this case, all the chicken gamma C receptors, and we take their sequence, because they're all amino acids. That's what proteins are made out of. We take their sequences and align them so that they're all the sequences are lined up. And each position in the sequence for one olfactory receptor, each position for this one, the equivalent in this one. They're all properly lined up. And then we take the most common amino acid at each position. Here's one position. The most common amino acid here is A, alanine. And we take that common one, and at the top, we put that towards our consensus sequence. Then we move to the next position. The most common amino acid there is C, cysteine. So our consensus sequence gets a cysteine, and so on until we've built an entire consensus olfactory receptor from start to finish. And this consensus receptor is a summation of all the receptors in the alignment, in this case, all the gamma C. Is everybody okay with that? So I think the take-home message is that we're testing many olfactory receptors with just one. So then once we have that consensus receptor, yeah.
Why do you take that approach to come up with something?
Yeah, this is because we use the mammal literature. We know this is very successful in mammals. And so we're taking the same techniques used for mammals and applying it to birds. So in mammals, when they want to test a mouse olfactory receptor or what it responds to, they do the same thing. They make a consensus sequence. Yeah, so, okay. We don't know why. It's very strange. So my boss has an idea why, but I don't think it makes any sense. He has a whole slide and a description. It makes no sense. Yeah. No, because his idea is that this consensus olfactory receptor represents a sequence of olfactory receptors earlier in evolution when there were fewer. The idea is that, can I even explain this idea that I don't believe in? Like the problem is olfactory receptors are difficult to get to the plasma membrane. There's difficulty trafficking them, for the cell to traffic them to the correct location. For whatever reason, these consensus sequences traffic better, and we don't really know why. But that's one reason why we do it. The other reason being we can test a lot at once. Yeah.
So it's a non-genetic person.
Yeah.
Just make sure I'm understanding it right. So you're trying to prove that the olfactory receptors that you're seeing, these huge numbers of them, are actually being used for smell, not as signals from other organs.
Yes,
Is that the thing? Because you were saying ORs are used for all kinds of things in your body,
Yes.
not just for that
Yes.
stuff.
And so the goal here is to get these olfactory receptors to actually respond to a smell. Yeah.
That's what I was hoping for.
That's where we're going. Yeah. Yeah. So, but I'm going over the methods anyway. Just because I like to put us all through this punishment here. So okay, so once we have the consensus receptor, do you have a question? Do you want to say something, Sarah? Okay, cool. Seriously, interrupt me, like forget about the debate. Just like interrupt me anytime. So then we take this consensus receptor that we have now made from the alignment, and we're going to put it into cell culture, into mammal cells. So unfortunately, we're not getting easier here for you guys, but I'm going to try and walk you through it. So this right here is a 96 well plate. It's a plate with a 12x8 grid on it for 96 wells. In each one of those 96 wells, on the bottom, cells are growing. Mammal cells, human cells. And we can put that consensus olfactory receptor that we made into the mammal cells, and they will make it and create it just like they would any of their own proteins. And so we put the consensus olfactory receptor sequence into the mammal cells, and they make it, and then they put it on their plasma membranes, and they're in 96 wells. And so we can put a different odor potentially in each one of those 96 wells, and we can test hundreds of odors against these olfactory receptors to see what odors they respond to. And so if they do respond, the odor in black there will bind to the olfactory receptor, and that'll cause a cascade effect where this little thing here, cyclic AMP, is generated. Don't need to know about it. You just need to know that our assay measures the amount of cyclic AMP generated. Okay, so that's our proxy for the olfactory receptor being activated. If the olfactory receptor is not activated, if that black odor was not in the olfactory receptor, there would be no cyclic AMP generated. So if it binds, there is cyclic AMP. If it doesn't bind, there won't be any. This is probably the most involved slide. I think it gets better. It's downhill from. So this is exactly what we did. We took the consensus sequence, which is represented by that yellow circle. So everything downstream in the phylogeny of that point went towards the chicken gamma C consensus sequence. And we tested this consensus sequence against many hundreds of odors, like I said before. And we found a particular group of odors called pyrazines, which smell like green peppers, caused a response. So there's the chemical structure of these two pyrazines. When we do this experiment, the whole lab smells like green peppers. So kind of bothers anyone who isn't into that. But on the x-axis, the way you read this is the x-axis goes from low concentration of the odor to high concentration. And the y-axis is the response of the olfactory receptor relative to a control group of cells that has no olfactory receptor in it at all, just for a baseline comparison. And so you can see that as the concentration of these pyrazines increase, the response of the olfactory receptor relative to the negative control increases, showing that these consensus sequences are responding to these pyrazine molecules, these green pepper smells. Now, that was for the chicken consensus sequence of all gamma C, but then we wanted to narrow it down a little bit further. So then we took 4 more consensus sequences, which are from subsets of the chicken gamma C. So we have 1, which is blue, 2, which is green, 3, which is red, and 4, which is within 3, which is pink or purplish. And then we tested all those against the pyrazines. And remember, 2 is green there. We found that of those 4, 1 is a little strange with the response, but the really nice curve going from low concentration to high concentration is in green, that subclade, or that subgroup 2. So we now are very interested in this consensus for the subgroup 2. But the thing is, like, this result was nice and everything, and we liked it and all that, but the consensus sequence isn't real. It's not an actual thing that exists in a chicken in nature. We just made it up. We just invented it. We just ordered it from this company called IDT. So now we're done with consensus sequences. We're done ordering from a factory. Now we're going to go directly to chicken genomic DNA, and we're going to get actual native endogenous chicken olfactory receptors from the genomic DNA and test those. And when we did that, we cloned up like dozens and dozens of these, and we finally got one chicken native olfactory receptor within clade 2 that, with increasing concentration of the pyrazine, shows an increasing response relative to the control. And this is the first ever match of a bird olfactory receptor to an odor, and strongly suggests that the chicken can perceive what we perceive as a green pepper smell. And this pyrazine is used in pheromone communication in many different insect orders. So we're not exactly sure why the chicken uses this odor smell, but we suspect it could be involved in foraging, and that's a future direction that we're going to look at. And since then, we found 3 native or endogenous olfactory receptors that respond to these pyrazines, including this 2-methyloxy-3-isobutylpyrazine, which was shown in this paper from 22 years ago, that when they let chickens sniff this pyrazine, it caused the chickens to lay bigger eggs. So this paper I never heard of this journal, Zoological London, and this paper has been cited like 3 times in 22. I take it with a grain of salt, but maybe there's something interesting here for the poultry community to look into. And then you get the bigger egg. So that was the gamma C. So yes, the chicken gamma C respond to these pyrazine odors, but then we also wanted to test the alpha receptors as well. And so you remember chicken gamma C, they're only in birds. They're not in other organisms. We don't have an equivalent in other organisms, but that's not true of alpha and gamma. Alpha and gamma are shared across all the vertebrates. And like I've been saying, the mammal olfaction literature isn't hindered by this previous bias dogma. They're way far ahead of birds. So now we can leverage the mammal research for birds with these receptors that are shared. So this olfactory receptor, OR51E2, is a model olfactory receptor. It's our lab's favorite olfactory receptor. Our lab published the structure of this olfactory receptor last year in Nature. And so we know everything about this olfactory receptor. We know the whole structure, the whole sequence, how the odor comes into the binding pocket, which parts of the sequence interact with the odor, the orientation of the odor, how long the odor stays in, all this. So it's a great model. And so this again here is the chicken olfactory receptor phylogeny that we've been looking at before, except this is a circular version, but it's the same thing. And when you take the human OR51E2 and put it onto the chicken olfactory receptor phylogeny, so these are all chicken olfactory receptors with one human OR51E2 alpha receptor added, which is in red there. You see that when you add the human OR51E2, it forms a nice 1-to-1 relationship with a single chicken olfactory receptor, which we're calling chicken OR51E2. And so we know that mammals have an OR51E2, and we know that it binds to propionate and acetate. We know that chicken has an OR51E2. So then the natural question is, across 320 million years of independent vertebrate evolution between the last common ancestor of birds and mammals, will the odors be the same? And so when we do this test, we recapitulate or repeat the result of the human OR51E2 responding to acetate and propionate with increasing concentration relative to the control. And we see the same result for the chicken OR51E2 with increasing concentration relative to the control, showing the preservation of olfactory receptor function across vertebrate evolution, across vertebrate classes. So yes, so do the alpha respond to odors? Yes, the OR51E2 ligands are preserved across different vertebrates. And finally, can we use morphology or ecology to predict the olfactory receptor counts? So the first thing we're going to do is look at morphology for the olfactory bulb. So the olfactory bulb, so after the olfactory receptor binds to the odor, downstream of that, there's a processing part of the brain for the odorant input, and that occurs in the structure called the olfactory bulb. And so here, this is the olfactory bulbs of a black vulture on top and a turkey vulture below. And the olfactory bulb is often, the size of it is often associated with the reliance of that species on smell. So you can see that the black vulture's olfactory bulb is much smaller than the turkey vulture's olfactory bulb. We know turkey vulture more heavily relies on smell than the black vulture. So this is sort of a positive control that has been shown in other animals to be that the olfactory bulb size is related to the olfactory receptor genomic counts. So we also did that in birds, and we saw a significant positive correlation even when correcting for phylogeny where the birds with the largest olfactory receptor counts up there, for example, the emu and the Takahi rail there and the nightjar, also had the largest olfactory bulbs, and the birds with the smallest counts of all, such as songbirds and the crow, also had the smallest olfactory bulbs relative to the size of the rest of the brain, or another part of the brain. And then we can also use ecological variables to compare with olfactory receptor counts. And so in the subset of the species that we measured, this is a 70 species subset, and across the subset we have 4 gains of nocturnality across the phylogeny in the kakapo, nocturnal parrot, the barn owl, the black rail, and these 2 nightjars. And we see that after correcting for phylogeny, there is a significant increase in the number of olfactory receptors in the genome for nocturnal birds compared to diurnal birds, showing that nocturnality may have an effect on the total olfactory receptor counts in the genome. And then we didn't, so that this was sort of a visual versus olfaction trade-off that we were looking for. And we also looked for an auditory olfaction trade-off. We didn't see that a comparison between song learning birds and birds with an innate song showed a difference in olfactory receptor repertoires, but we did show that for the alpha subgroup of olfactory receptors, there were more alpha olfactory receptors in the birds with innate songs compared to the birds that learn their songs across those 3 gains in hummingbirds, parrots, and songbirds. So not sure exactly what that means specifically for alpha olfactory receptors. But so can morphology and ecology predict the olfactory receptor counts? Yes, for olfactory bulb size and nocturnality. And so in the future, we're going to perform more odor exposure and behavioral tests, particularly on the chicken, to figure out what that pyrazine is doing behaviorally. And we're also going to expand these methods that we've applied to the chicken to diverse orders of birds, such as turkey vulture and the honey guide. And also we have some exciting ant bird stuff working on how they detect the ants. And so just thank my lab, Hiro Matsunami lab, everybody in the lab, Chris Balakrishnan, my PhD advisor, and all the funding and all that. And that's it. Yeah.
My question's on sensitivity. So we have different, we know that different noses can pick up small amounts of odor molecules versus in some larger amounts.
Yeah.
Does that happen, and how does that relate to it?
Well, yeah, it has to do with how well what kind of affinity the odor has for the binding pocket of the olfactory receptor. But you could imagine that different birds need a different amount. Like the turkey vulture, when it's way high up in the sky, would need an incredible sensitivity. But then it also must have to augment that a little bit, because then when it flies down, it's right next to the carcass. If it was the same signal input, it would be overwhelming to the turkey vulture. So yeah, there's a lot of variability with that. And we see that in the assay where different olfactory receptors in mammals, where different olfactory receptors will respond to the same odor, but at very different concentrations.
How did you get interested in bird smells?
Yeah, in my neuro undergraduate class, they had us read this Linda Buck paper where they where they first discovered the olfactory receptors in mammals, and I thought that was really cool. And then I started thinking about vultures, and now I'm here giving away God of honor. So. Yeah. Oh, I'm sorry. I got to repeat the question. Sorry.
You know, anyone's tried to repeat Darwin's experiment.
Do I know if anyone's tried to repeat Darwin's experiment? No, I don't. But that would be really fun. If I'm ever in front of a row of chained Andean condors, I absolutely will go to a deli and okay. Yeah, I think it would be interesting to repeat that with a raptor or some bird that looks for meat. Yeah. Yeah.
Questions in the chat.
Oh, uh, are you adding the chemical or just the odor to the wells? Yeah, so the chemical, the odor is added to the odor is first diluted in ethanol and then it's diluted in a separate buffer, which we call HBSS. But those buffers that it's in are meant to not have any response to the olfactory receptor. It's just the odor that would be responsible for the response. And then we also have a no odor to control just in case to measure for that. I think that's it.
Robert, I have a
Yeah.
question. So first of all, that was fascinating.
Yes.
Thank you. Um, you were noting that some of these bird species, the kiwi in particular, have far more olfactory receptors. And is that like types of receptors than we have? Or
Yeah, types. Yeah. Mhm.
And so what are like the implications of that? Does that mean that in theory they could smell a larger variety of smells than other animals, or or do you really know?
Yeah,
You
so
don't expect it.
I think so a paper did come out recent. So the question is, if you have more olfactory receptors in your genome, what does that mean exactly for smell, right? So there was a paper that came out which said that concluded that the more olfactory receptors you have, the more types of smells that you can detect. But I don't necessarily believe that result or think that that's always the case. We don't have proof of that this huge diversity of thousands of receptors in the kiwi are used for detecting all sorts of different odors. We're not sure. And conversely, the honey guide indicator has actually a pretty low count of receptors, but and we haven't been able to get the honey guide receptors to respond to anything. But uh, from my understanding, the honey guide is just looking for wax and it's just trying to find wax from these beehives. So if it's cued into a particular compound, it may not need a whole host of olfactory receptors to be very sensitive to that one compound and rely on it. So I think there's a little bit of a mismatch between the number of olfactory receptors and the reliance of that species on smell. Yeah.
I have a similar question about the
Mhm.
gamma C.
Yeah.
So you were saying that the birds have a lot of like essentially unique olfactory receptors.
Yeah.
Like, what would you theorize like that is reflective of?
Yeah, we don't know. That's what we're trying to figure out. So we didn't even know if they were involved in smell, and now it's looking like they are based on their distribution. But the other strange thing is they evolve in a way where they group based on species. So all of the chicken gamaceae are more closely related to each other than all of the zebrafinch gamaceae. Like the as opposed to a traditional gene, which are more closely related uh, to like between species than between other genes. So all of the chicken gamaceae form a single clade and all of the zebrafinch gamaceae will form a single clade. And so the different species have different gamaceae sequences that are unique to that species, but we still don't know why or what's going on with it. Yeah.
When you say there is a close back between the human olfactory receptors, they could be food.
Yeah, that's right. So yeah, so the question is, like I said, that a chicken receptor was similar to the human receptor. It's basically like their sequence. So their A, T, C's, and G's, like the sequence of that chicken receptor, of that human receptor, is closer to that chicken receptor. Or rather, the chicken receptor is more similar to the human receptor than it is to any other chicken receptor. Yeah.
It seems to me that you, in a way, remove the speciation from the picture, and you just look at the sequence. There can be a sequence for a chicken that we're doing. Has there been a study to look at how a particular sequence responds to certain odors? To let the lookup table, this sequence looks like this, and probably respond this way.
Yeah, so that's probably so the question is there any way to predict based on sequence alone what the olfactory receptor binds to? Is that? Yeah, so that is probably the most active area of chemosensory biology right now, is trying to model the just based on the sequence, to create a model on the computer and predict what odors will bind. Yep, it's active, and our lab is trying to get on that train. So
Okay, there's
Yeah.
two more questions.
okay. Oh, and then you guys. Oh, okay, perfect. I've always returned baby birds to their nests and fallen out. Parents can't smell their babies on the. Yeah, so not to dispel, like, there's multiple myths going on here. So right, you can return the baby back to the nest and it's not a problem. That's one myth, that you don't want to return, or that you don't want to disturb the nest at all, or like if you disturb the nest, the parents will abandon it immediately, even though they put in so much work to create that nest and over. Anyway, so but then to say that when you replace the baby that they won't smell the baby, that we don't know. Like, my research can't prove that's true one way or another. But the behavioral studies in the zebrafinch have shown kin recognition, where the parents can distinguish between their babies and other babies. So based on that one study, I would think that the birds can smell their babies and maybe know that your, I don't know, hand was on it or something. I'm not sure, but I'm pro bird smell, so I would believe that they could notice that. And then if birds can detect the smell of their own eggs, yeah, wow, what a great question. Fantastic question. Yeah, is the cowbird using some, sorry, if birds can detect the smell of their own eggs, why do birds get fooled into hatching the eggs of other bird species? So maybe the cowbird has some olfactory signal, or cuckoo has some olfactory signal that it's mimicking when it lays that egg. I never considered that. That's a really great question. That could be an entire dissertation there. Yeah.
Mentioning research on zebra finches, and you mentioned there's corridors in there having therapeutic types of receptors. Pass range in general, what are the general patterns and differences? Which groups have more and which have fewer patterns?
Yeah, so passerines are so neo-aves. So the if you do an ancestral state to look at the evolution of the counts over time. Yeah, so the ancestor of all birds, presumably and presumably a theropod dinosaur, has very high counts of olfactory receptors, and those high counts are maintained in the ratite and paleognathy lineage, as well as the fowl, galloansary, duck, and chicken lineage. Those counts are high. But then when Neo-Aves diverges around the Cretaceous extinction, there's a major loss of olfactory receptors, and those lineages all tend to have less. And then for passerines are typically low, but there are a few select species which have very high counts. So of all the passerines, the highest tend to be New World sparrows. So New World sparrows and Amospiza and Passarella, I think, is the other one that we've measured. We have a bunch of New World sparrows in there, but they have high counts. Yellow warbler was one that had very high counts. And then icterids are a little bit variable, but red-winged blackbird had very high counts. in its genome. So it's a little bit all over, but the general trend in passerines is that it's lower. I tried to correlate. So those ecological variables at the end where I had nocturnality and song learning, I tried to correlate all sorts of different habitat type, diet, all this, but nothing came out, at least for my data. So, yeah.
Alright, well, any other questions? One last one? All right, join me in thanking Robert again.
Thank you. Thank you. Thank you. Appreciate it.
All right, well, that is our program for this evening. There's some snacks in the back.
Great, thanks everyone. Bye.